All right, so the topic for this video is going to be the process of gene transfer and ways that DNA can be transferred between bacteria. Because what we've talked about previously, we talked about bacteria undergoing binary fission and giving rise to two identical cells. So they've got all the same genetic material. But there is a way to introduce additional genes or genetic material into bacteria, because bacteria don't mate per se. They don't have sexual reproduction like eukaryotic organisms do. All right? Typically what's going to happen with gene transfer, most often the genes in question are going to be on a separate piece of DNA from the genome associated with the bacteria. And that separate piece of DNA is the plasmid. Okay? Plasmid is a circular bit of DNA. I should draw this because it is double-stranded, so we'll make it look kind of double-stranded. All right? So with that genetic material, it's going to transfer to the bacteria, typically on a plasmid, although you can get genes that transfer just in bits and pieces as well. So you can have bits of chromosomes that are picked up by the bacteria, and that can be taken into their bacterial genome. All right? But most often it's via a plasmid. And with gene transfer, there is an advantage typically that that bit of DNA is going to impart on the bacteria. All right? The most common one, and I'm sure you have at some point seen something in the news about the rise of antibiotic resistance with bacteria. And this is a big problem in the medical community because people come in with bacterial infections, and the thing about bacteria is they've come up with ways to get around that antibiotic. So you take that antibiotic, and even though it's present in your system, the bacteria are still able to multiply because they have inherited genes that are associated with antibiotic resistance. So the bacteria are able to grow even though antibiotic is present. Probably the one you've heard the most about is going to be MRSA, all right? And MRSA stands for methicillin-resistant Staphylococcus aureus. So the bacteria that has developed the resistance is Staphylococcus aureus, and the methicillin is the particular antibiotic that it has acquired the resistance to. All right? Staph aureus actually is pretty good at picking up antibiotic resistance along the way. Staph aureus became resistant to the antibiotic penicillin way back in the 1940s. So antibiotic resistance, you may think it's something new, but actually antibiotic resistance has been around for a long time. Thing is, we've developed newer antibiotics, and well, if it's resistant to this one, we'll just throw another one at it. But that's kind of snowballed over time. So now we're on the limiting end of not having new antibiotics that are being developed and more resistance that's happening in bacteria. So what happens, a plasmid gets transferred to the bacteria. That causes antibiotic resistance. All right? So that's a big one. Second thing that can be transferred are going to be virulence factors. All right? Those virulence factors are going to confer genes that are going to result in the bacteria being pathogenic. All right? And if it's pathogenic, we're talking about something that causes disease in humans. But again, it offers an advantage for the bacteria in survival. And that advantage is if it can get past your immune system, that allows that bacteria to still replicate and generate more offspring. And that's really the goal, all right? So with gene transfer, what all of these are going to have in common is that that bit of DNA that is transferred from one bacteria to the other confers some kind of positive characteristic for the bacteria, whether it be able to grow in the presence of antibiotics, whether it be virulence factors, which make it able to infect people, all right? The other mode or advantage that can be conferred genetically, again, gets to resistance. And in this case, it's resistance to heavy metals. So with the heavy metals, we're not talking like heavy metal hairbands, so we're not going Metallica or Poison. We're talking heavy metals as far as in the environment. So with bacteria, all right, bacteria don't grow well, normally anyway. I need to get a new orange marker. The bacteria don't grow well on metal surfaces. This is why in surgical suites you've got so much stainless steel, because bacteria tend to not want to grow on the stainless steel. That keeps bacterial loads low in surgical suites, which is advantageous if you're having surgery because you don't want to introduce pathogens into the body during surgery. But there are bacteria, again, through gene transfer that can inherit little bits of DNA on plasmids that allow them then to grow in the presence of a metal, okay? Now with gene transfer, there will be a donor and a recipient, okay? So the donor is going to be the one that has the plasmid. The recipient is going to inherit the plasmid in this process, all right? And with the process of gene transfer, you're probably thinking, well, great, well, let's just take that out and not let bacteria do that. Let's not let them transfer DNA. There is an advantage that we use gene transfer for with bacteria. And what that is that allows us to use bacteria to make proteins that we want. And an example of how we use bacteria to do this is with the protein, human protein insulin. Most insulin now is what's known as recombinant, and that's something else that will come up in this unit. All right, when we're talking about recombinant, you're talking about combining DNA from two different organisms, all right? So you can have recombinant DNA between two bacteria. You can have recombinant DNA between bacteria and humans. When we artificially introduce the gene for insulin into bacteria, essentially we're taking advantage of the transcription and translation that bacteria undergo to make that protein for us. And what that does is that allows a much more uniform production of insulin. So the insulin is more reliable, the dose is more dependable, and it doesn't, because it's bacteria, you don't have to worry about introducing disease from another person, all right? So we really can use bacteria and the process of gene transfer to our advantage. The next video I'm going to talk about the types of gene transfer, and we're going to have 4 types of gene transfer that we're going to go over, all right? So thank you for your attention. Let me know if you have any questions.